Okay. So good evening, everybody. I apologize for the postponement of the session. So that is why I am seeing the attendance is also significantly low. Ideally, I'll be taking these courses every Monday from 7 to 9. This Monday, I had some unavoidable core that I needed to complete, due to which I couldn't take the session, right? But I'll upload this one, which because of lack of attendance, it will be more of a recorded session, I guess. So I'll just go through the material that I have and I'll upload it on YouTube for you to uh, go through it, right? Without wasting time, I'll just share my screen first, right? So, yeah. So first, what we can go through is the previous year questions from week 0 and week 1 that were given in your course. And then for the latter part of the session, what I have kept is basics of molecular biology, because there were a lot of basics that were introduced during the first week of lectures. So we have not fully dived into the neuroscience part as yet. But there were a lot of concepts that we have gone through, right, in molecular biology. Because we have time over here, we will go through them in a little bit more detail. We'll still not delve in deep into that because it is not a molecular biology class, but we'll still try to cover a little more so that it refreshes your memory, right? For instance, what happened in the Michelson-Stahl experiment, what exactly happened in the Hershey-Chase experiment, and so on, right? But that will be the latter part of the session. First, what we'll be doing is we'll go through the previous year questions for week 0 and week 1. And Week 0 questions are an amalgamation from the entire course. So by the time you are through with this course, you will know the answers to most of those questions, right? That is our aim. So even if you don't know it at this point, it is okay. This is what the course is for. As and when you read more, you study more, you will understand more about the this subject, neuroscience, neurobiology, right? And then you'll be able to solve it better. So for now, let us just begin with this, right? Yeah, the first question, for instance, is the cell body of a neuron is also referred to as, the options that were given were axon, dendrite, soma, or synapse, right? So even if you are very, very new to biology itself, maybe you don't know the answer to this. But if you have studied neuron as a cell type, even in your cell biology or molecular biology courses, you will be easily able to answer this, right? We'll see what the parts of the neuron are. The answer for this one is soma. Soma itself means cell body, right? Axon is that long kind of fiber that is coming out of the cell body of the neuron. Dendrites are those tree-like or root-like structures, as you can see in this background, these things that are coming out. And synapse is basically the space between two neurons, right, where the transmission of signal occurs. I have also kept another representation of the neuron. As you can see over here, this part, this core part is the cell body, which is also referred to as soma, right? This part is the axon. Sometimes it's myelinated, sometimes it's not myelinated. We'll study about what the importance of this myelination is, or why sometimes it is not myelinated, right? And eventually we will come on to the other parts as well. Okay. So, moving on to the next question, it is action potentials are transmitted through to the cell body of the neuron from other neurons through which of these? Through axons, through dendrites, through nodes of ganglia, or through the soma or the cell body, right? So, again, we can visit the same structure and we can see that dendrites, these root-like structures that are arising out of the cell body or the soma, are the ones that actually receive signal from another neuron, right? So this is one neuron, which is a presynaptic neuron, which means this is the neuron that is passing on the signal, and postsynaptic neuron is the one that is receiving that signal, right? So the receiving end will be the part where the soma is, and which particular part will actually receive the signal? It is these dendrites, right? So that is the answer for this one. The synapse where this exchange of signal is taking place has also been zoomed in over here for your reference, and we'll study it in more detail as and when we go through the course. Yeah. So the other question is the resting membrane potential of a nerve cell is, right? So again, it is also sometimes an aptitude-based question in a lot of competitive exams, JAM, GATB, GATE, CSIR, right? So the correct answer to this is minus 70 millivolt, right? And we will, again, throughout this course, what we will understand is we know, often we do know this answer, but we don't know how this minus 70 millivolt is actually achieved or why it is this number only, right? So that we will understand over here, that it is basically an average of the different kinds of membrane potentials for different ions. It is an average value out of all of those, right? So eventually we'll study that as well. As you can see over here, minus 70 millivolt is what the resting membrane potential generally is. Then, why I did not stay in the last image for very long was because it had the answer to this question also, right? So we'll see it again in a few seconds, that the extracellular fluid has a higher concentration of dash, while the intracellular fluid has a higher concentration of what, right? So these are your options for extracellular versus intracellular. It could either be sodium and potassium, or potassium and sodium, chloride and potassium, or potassium and chloride, right? So this again has nothing much to do, or it is not very specific to neurobiology per se, right? Even in your other cell types, this is what is generally being followed, right? So extracellular fluid generally has a higher concentration of sodium, while the intracellular fluid has a higher concentration of potassium, right? That is why there is a presence of that active transport system, also Na plus K plus, which maintains this balance, right? The sodium potassium pump, as we generally call it, right? And these are the most common uh, ions that are present, right? So over here you can see that sodium in the extracellular fluid, this is the value, while intracellular is much less than that. In case of potassium, it is the almost the opposite, right? Extracellular fluid has much more than the uh, intracellular has much more than the extracellular fluid, right? Similarly, extracellular fluid also has higher amount of calcium. It also has higher amount of chloride ions, right? And many others, right? In the previous picture also, if we go, just a moment, over here you can see how it is represented is also telling you what the answer to that question is, right? Over here outside, you can see more of these pink molecules that are the Na molecules that are present, and K plus, that is the blue colored molecules over here, they are more in number inside, or the cytoplasm or the intracellular fluid, right? Uh, yeah. Then the next question is that the nodes of Ranvier are located in the cell body, or along the axon of the neuron, or the dendrites, or both the cell body and the axon, right? So the correct answer to this is that it is located along the axon only, right? Why it is only located along the axon? Because these nodes of Ranvier are basically these openings where myelin sheath is not covering the axon, right? So, in myelinated axons, there are openings at a regular frequency. These openings are known as nodes of Ranvier. And since there is no myelination itself in the cell body or the dendrites, we don't have nodes of Ranvier over there, right? Then the next one is neurotransmitters relay signals across one, the synaptic gap or the axon gap or the cell body or the node of Ranvier, right? So, which one among this is the correct answer? So, in this particular case, it is the synaptic gap, right? Synapse itself, as we were discussing in this particular figure earlier also, synapse is the region where the exchange of information is taking place, right? Between this presynaptic neuron and this postsynaptic neuron. So, this is the area where the neurotransmitters which are helping to relay this information are actually present and they are relaying signals from this neuron to this neuron, right? Thereafter, we ask another question. The central nervous system consists of which of these, right? Is it the brain and the spinal cord, or the brain and the cerebellum, or the cerebellum and brainstem, or spinal cord as well as cerebellum, right? One second. Yeah. So the correct answer to this is brain and spinal cord, right? Why? Because PNS actually consists of these two, brain and spinal cord. The other parts, other neurons that are outside or nerves that are outside of this are a part of the peripheral nervous system or the PNS. And over here, the options that are given to you apart from this is brainstem and cerebellum, right? Both cerebellum as well as brainstem are a part of the brain itself, right? It is not a separate entity. So that is why this is the most appropriate answer. So as you can see over here, this is the central nervous system, the brain and the spinal cord, while the peripheral nervous system consists of all the other nerves that are innervating the body, right? Then which brain lobe is most involved in visual processing? The frontal lobe, parietal lobe, occipital lobe, or temporal lobe. Now, this is a typical neuroscience question. So, since you have still not started understanding this neuroscience part as yet, right? I have kept this representation of the brain to first tell you what these lobes are, right? Because earlier what we used to study was the hindbrain, midbrain, forebrain, like that, cerebellum, brainstem, right? That is all we used to study. But over here, you can see that the forebrain, for instance, or the cerebrum, for instance, is also divided into multiple lobes. These four are the main lobes, right? The frontal lobe, the parietal lobe, occipital lobe, as well as the temporal lobe. Apart from that, cerebellum is there, and the spinal cord or brainstem region is present as and represented in the blue color. Right. So these are the various lobes. And you will study about this in your course. Right. Now, up in all of these, which one particular lobe is actually involved in the visual processing? Right. So the answer for that, which you will also be able to answer it very in a few weeks, is occipital lobe, right? This particular region. So this part I have kept another representation which gives the major functions of each of these lobes, right? So frontal lobe, for instance, is responsible for decision making, from for problem solving, for major emotions and behavior regulation, speech production, etc. Right? The parietal lobe is responsible for processing of senses, spatial awareness, understanding where our body is in space. Occipital, the one that is we have listed over here, is actually the major vision and visual processing area, right? Apart from this, there is temporal lobe, which is the place where memory formation, language understanding, as well as hearing occurs, right? And we have also given certain other oh, it is just a repetition of what is written over here. Cerebellum is involved in balance and coordination. You should listen to this because there is a question related to this also in this particular presentation itself. Thereafter, there is brainstem, which controls functions such as breathing, heart rate, and blood pressure, as well as sleep cycles. Right. So for now, we shift to again the cellular part of neuroscience, which are the cells that are responsible for producing myelin sheath around the neurons, right? So are these osteocytes or are these Schwann cells, oligodendrocytes, or both Schwann cells as well as oligodendrocytes, right? So the first one is osteocyte, right? The first option that is given is osteocyte. So you might already be aware, or you might know the etiology of this word osteo. It has something to do with bone, right? So anyway, you can rule this out. That osteocyte will not be the answer because it is not producing myelin. It is generating those bone structures, bone cells, right? So the other two options were Schwann cells and oligodendrocytes. This you may or may not have heard of before, but both of them are actually involved in producing this myelin sheath. Schwann cells help in the production of myelin sheath in the peripheral nervous system, while oligodendrocytes help in production of myelin sheath within the central nervous system. Right. So both these cells help in producing this myelin sheath. Thereafter, the next question is neurotransmitters travel from the presynaptic membrane to the postsynaptic membrane via which of these processes, right? Is it based on osmosis or diffusion or active transport or reverse osmosis, right? Over here, the answer is simple. There is no semi-permeable membrane or there's no membrane per se that is present within this synapse region, right? And there are no active transporters that are also present, right? So it is simply through diffusion that this process takes place. Thereafter, the other question is which condition results from the death of dopamine producing neurons in the brain? So these neurons are also known as dopaminergic neurons. Dopamine, as you know, is a primary is one of the primary neurotransmitters in the brain. It is involved in a lot of kinds of signal processing, right? It could be related to the mood elevation also, and it could also be related to the motor functions, right? Those kinds of signals also, dopamine producing neurons, like dopamine as a neurotransmitter, it performs, right? And these are certain diseases that are given over here. It is multiple sclerosis, seizure disorder, Parkinson's disease, and stroke, right? So out of these, actually, Parkinson's disease is well linked with death of these dopaminergic neurons, right? So over here, you can see a representation where this is a normal neuron, wherein the presynaptic neuron, which is the dopaminergic neuron, is able to release. So dopaminergic neuron is again a dopamine producing neuron, right? It is it has large reservoirs of dopamine within it, which it releases at the synapse, and uh, the receptors in the postsynaptic neuron are able to perceive this neurotransmitter, and it affects, or it is involved in normal movement or muscle function, right? While if there are Lewy bodies, which are like protein aggregates of some sorts, if they are present or they are affecting these dopaminergic neurons, then what happens is a condition known as Parkinson's, wherein there is very less production or release of these dopamine neurotransmitters in the synapse region, and thus the movement disorders occur, which is a key symptom of uh, Parkinson's disease, right? Please give me a moment. So I think I'll reshare the screen once. Okay. Uh, so if my cold doesn't get better, I think I will stop the class prematurely and I'll cover it in the upcoming session on Monday, right? So depending on how I long I can sustain, I'll be able to teach. Yeah. So the next question is, how do neurons transmit signals to each other, right? So do they do this via electrical signals or chemical signals or through impulses or through both electrical and chemical signals, right? So how this happens is through actually both the electrical and chemical signals. There are both kinds of synapses that are present. Sorry. There are electrical synapses also and chemical synapses also. There are more number of chemical synapses that are actually present. But as you would expect, the signal transmission will be more rapid in case of electrical synapses, right? Because the rate of flow of ions over here will be much more than the flow of ions when a synapse is present and through this means, right? But both the kinds of signal transmission actually occurs. Uh, Give me a moment. I just apply some balm. I'll share again. Yeah. So the next question is, damage to which brain region leads to loss of balance, coordination, as well as decreased muscle tone, right? So is it parietal lobe or thalamus or cerebellum or occipital lobe? So this even in our class 10th and 11th books we have studied that which is the organ that is responsible for this balance and coordination, and a while back also I told you to pay attention when I was describing those various lobes of the brain and their function, right? So what is the answer for this one? It is basically cerebellum, right? So over here you can see cerebellum is the region that is responsible for balance as well as coordination. Then Broca's area is located in which of the following? Frontal lobe, parietal lobe, temporal lobe, or occipital lobe. Now, before we understand which area of the brain it is located in, let us understand what exactly is Broca's area, right? So Broca's area is one of the areas which is responsible for speech production, basically, right? So what it means is that there was a patient in which this particular area of the brain was not functioning, or it was damaged through some way. Maybe there was some epilepsy, it was an epileptic focal point, or it was damaged during an accident. That part I don't recall very well, but it was this area of the brain that was damaged somehow. And due to that, what used to happen was the person was able to understand what the other person is trying to communicate. He could process that. He could, in his mind, he could form what has to be said. But when he used to start saying that thing, the speech wouldn't come out properly, right? The speech production wasn't happening, right? So the articulation of the production of the speech was what was affected. So that region came to be known as Broca's area. There is a contrary to this, there is another area in the brain which is located in another region which is known as Wernicke's area. Now, in that area, what used to happen was in another patient, that particular region was damaged, right? So, what used to happen is the person couldn't understand what was being said to him, the person couldn't comprehend what is being said, but the person could speak, right? But because that person can't understand what is being communicated to him or can't articulate what has to be said, he used to speak clearly, but whatever he used to speak would make no sense at all, right? Because that person has lost the skill of communication or articulation. The person can speak, but that is the skill that is lost. In Broca's area, the communication is intact, but the uh, uh, ability to produce words is what is lost, right? So and these were present in two very far apart areas. So that is how we also came to know that speech and other senses, they are basically functioning through various regions of the brain. It is not just located in one region of the brain, or one region of the brain might not be responsible for one sole purpose. Right? It might be different regions of the brain that are coming together and helping perform one simple function also, right? So Broca's area per se is located in the frontal lobe. You can see this is the Broca's area that has been highlighted over here. It is referred to as the anterior speech area, and this is the region where it is present, right? The frontal lobe area. The other area that I was talking about is this Wernicke's area, right? This is the Wernicke's area. It is present somewhere in this parietal lobe, right? Yeah. So speech articulation is one thing, speech production is one thing. All these things we came to know through these kinds of studies, right? Or reports from patients. Then what is the primary effect of myelination on axons, right? Does it protect them from damage? It slows the signal propagation. It prevents crosstalk between axons, or it greatly increases signal conduction speed, right? So which of these is actually true? It basically greatly increases the speed of signal conduction, right? So what you can imagine is this myelination is kind of like an insulation that is present, right? So if you take a wire that is not insulated properly, it will have slower speed of conduction of electrons, while if there is a wire that that is well insulated, the speed of conduction of ions electrons will increase. Why? Because over here loss is not happening outside, right? But in the naked wire, the loss will happen from while traveling, right? So that is what is the main effect of myelination on axons, right? So we hear what effect unmyelinated axon, what happens? The signal travels throughout the axon, right? In myelinated axon, what happens? It appears because it is insulated in between. It appears as the signal is jumping from one node of Ranvier to the other, right? Because within this myelinated region, there is no decay that is happening at all, or very minimal decay that is happening, right? So it appears as if signal is jumping from here to here, right? While if it is not myelinated, it will continuously decay like while propagating. Okay. Then from here comes the actual week one lectures that you have done, questions related to that, right? So, for instance, which historical period is highlighted for contribution to neuroscience? The answer to this one is 1940s and 50s, right? This was the title of one of your lectures also, who is credited with laying the foundation for chromosome theory of inheritance, right? So, which of these great scientists is, credited with laying foundation for chromosome theory of inheritance, right? Is it Gregor Mendel or Thomas Hunt Morgan or Barbara McClinton or Frederick Griffith, right? So, the correct answer for this is Thomas Hunt Morgan out of the options that are listed over here. at least, right? Along with Morgan, Walter and Sutton had also a great role to, they also contributed a lot to the chromosome theory of inheritance. But out of these, Thomas Hunt Morgan is the correct answer. So, if the white eye gene was present, right? What he saw was like this is the chromosome theory of inheritance that like from a somatic cell, the chromosomes they replicate, right? After that, they undergo a meiosis one, wherein there is reduction of chromosome number, which where they are duplicated into daughter cells, and further these chromosomal arms also separate, and then there is a further reduction of chromosomes from diploid, they become haploid over here, and that is how these various chromosomes are transmitted, right? Over here, you can see capital Y, capital R, small r, right? So these are traits. So there will be few daughter cells in which there will be capital Y, capital R that will be transferred, few daughter cells wherein small y, small r will be transferred. Similarly, few where Y, capital this is homologous chromosome. So that is the combination that we get in F1 progeny at least, right? So what experiment Thomas Hunt Morgan did? So Thomas Hunt Morgan, if you remember, used to work on fruit flies, right? So what he saw was that if the Y gene was present, right, it would always expressed in males only because they have one copy of the gene. So it was probably present in the sex chromosome, right? The males had one copy of that X chromosome while the females had two copies of those X chromosomes. So in male, if that gene for white eye was present, it would always result in white eye in the male, right? On the other hand, if the females had that gene for a white eye, then it may or may not express because females have two copies of X chromosome, and this is a recessive trait, right? So only if it is present in two copies at least of the eye of the chromosomes of the female will it express. If it is not present, then it will not express, right? So that is why it was a recessive trait, right? Uh, this basically gave insights into chromosomal theory of inheritance, and later Walton Sutton also improved that theory and presented it to the world. Then another very simple question: which system is a part of the sensory division of the nervous system? Is it vision or posture or higher motor centers or neuroendocrine system? Right. So the correct answer for this one is vision. Then what structural features of DNA did Watson and Crick describe? Did they say that DNA was double helix with complementary base pairing, or that it was single helical, or it was a triple helix with random base pairing, or it was a helix with no defined structure, right? So if you know what DNA looks like or what is its property today, you will be easily able to answer this because Watson and Crick were the ones who correctly described the structural feature of DNA, which is that it is a double helix with complementary base pairing, right? So over here you can see this one is the DNA, this is the RNA. So DNA is this is what a single helix is, right? And DNA is a double helix, right? And it has complementary base pairing, right? So G is going to, form a bond with C. A is going to form a bond with T. So that is what complementarity is. Then what is the primary role of RNA in protein synthesis, right? So does it help in replication of DNA, or it acts as a template for protein synthesis, or it transports oxygen, or it stores genetic material, right? So we can rule out the options that we think are not correct, right? Is RNA responsible for replicating DNA? No, right? Polymerase is what is responsible for that. You know, transporting oxygen is again not a function of RNA, it is a function of a particular type of protein, right? Storing genetic material, in most of the higher organisms, that is not the case. In some lower organisms, it is actually the genetic material. But over here, the most accurate answer is acting as a template for protein synthesis. That is universal in all organisms, right? So, as per central dogma, we can the actual primitive central dogma used to say you can ignore these dashed processes, and what it used to say is that DNA forms RNA, RNA forms protein, and this process is irreversible. It is unidirectional only. It can only DNA can only form RNA, RNA can only form protein, right? And this step is not reversible. It is just unidirectional, right? But in either case, RNA does act as a template for protein synthesis, right? Then what is the significance of the specific pairing in DNA, which was proposed by Watson and Crick, right? So in their paper, in that last line of their paper, what did they say? That it explains the compact structure of the DNA, or it suggests a possible copying mechanism for the genetic material, or it determines orientation of RNA, or it determines, so, okay, this option is given twice, right? But the correct answer is it suggests a possible copying mechanism for the genetic material, right? Because it was double helical, it would probably dissociate, and one of the arms of the helix, it would act as a template for, further replication, or both of the arms could also act as templates for further replication, right? So that is the possible copying mechanism that they suggested. Then the next question is which statement aligns with the central dogma hypothesis, the original one, right? Information flows from RNA to DNA and then to proteins. Information flows from DNA to RNA and to protein and cannot reverse, or the information flows only from proteins to DNA, or DNA does not participate in the information transfer at all, right? So there's only one true statement over here, which is information flows from DNA to RNA to protein and cannot reverse. This was the original central dogma, right? This is not true at this current time. There are some modifications, right? Like reverse transcription or RNA itself acting as a protein, replication happening. These kinds of additions have been, or exceptions are present, right? Then the other question is, what is the discovery which was made by Hershey and Chase in 1952? Right? So, what is the discovery that they made? Whether DNA is the genetic material, or RNA plays a role in transcription, or DNA undergoes mutation, or chromosomes are inherited independently. Right? So the actual answer is that DNA is a genetic material. What was the Hershey-Chase experiment? They basically took a bacteria and a bacteriophage. What are bacteriophages? They are viruses that infect the bacteria, right? What they did was they labeled these bacteriophages. The bacteriophage has two major components. One is the protein coat that is present, right, which they labeled with sulfur, and the one was the material within these bacteriophages, which was DNA over here, which they labeled with phosphorus, right? And what they did was they let these labeled bacteriophages infect different populations of the bacteria, right? So once the sulfur labeled bacteriophages infected the population, what they did was they first, after infection, they blended so that whatever bacteriophage coating is remaining outside of the bacteria, it dissociates, or it goes away from the bacteria. Only whatever has gone into the bacteria will remain, right? After that, centrifugation was done to pellet down the bacterial cells, and what was seen was that there were no sulfur labeled cells that were seen over here, right? So all the sulfur labeled cells went in the supernatant, or they went away, right? So they said that okay, protein at least did not go inside, right? Then they repeated this experiment with phosphorus labeled DNA, and after centrifugation, what they found was that in these pelleted cells, they had phosphorus present, right? So, what they deciphered from this experiment was that indeed, the DNA was the material that was being transferred from this viral particles to the bacteria and it was infecting the bacteria, right? So this is the genetic material of the bacteriophage, which it needs to replicate within the machinery of the bacteria to survive and further, replicate. Then what is one structural limitation of ribose sugar that prevents its use in DNA, right? So ribose sugar is present in RNA, right? That is ribonucleic acid because it has a ribose sugar and DNA is deoxyribonucleic acid means it has a deoxyribose sugar, right? So the answer lies within this statement itself, right? So it cannot form bonds with bases. It its extra oxygen atom would create unfavorable van der Waals contacts. It lacks the ability to form helices or it does not bind phosphates, right? So what happens is the ribose sugar, right? It has an extra oxygen over here, right? Which the deoxyribose sugar doesn't have. This makes the ribose sugar more reactive, right? And it forms unfavorable van der Waals contacts that makes RNA less stable than DNA, right? So that is why it prevents its use in DNA. This is the final question, which is what genetic disorder is caused by a defect in the gene for hemoglobin, right? So the options that were provided were Down syndrome, Klinefelter syndrome, sickle cell anemia, or trisomy of. Okay, sorry. I'll just stop. I'll reshare the screen, right?